Everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Branching Out in the Area of Genetic Neurology, Collaborating with Australian Clinical Trials Experts. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and be made available to you for future streaming on xtalks.com. At this point, I'd like to thank Nucleus Network and Praxis Precision Medicines who developed the content for this presentation. Nucleus Network is the only multi-site phase 1 clinical trials provider located in Australia and the USA. They provide high-quality first-in-human and early-phase trials for biotechnology and pharmaceutical companies across the USA, Europe, and Asia. Located within cutting-edge health precincts, their cost-effective accelerated clinical development solutions are supported by advanced technology, clinical excellence, and research expertise. And now I will introduce Praxis Precision Medicine, which is leveraging important breakthroughs in the genetics of epilepsy and neuropsychiatric disorders to build a multi-product biotechnology company. Launched 3 years ago, Praxis is comprised of a world-class team of experienced senior managers, directors, and advisors. With greater than $100 million of capital raised to date from a syndicate of top-tier life science investors, Praxis is advancing a de-risked portfolio of highly differentiated clinical stage assets and a robust discovery pipeline spanning epilepsy and broad CNS indications. Now, it is my pleasure to introduce your speakers for today's event. And your first speaker is Dr. Gabi Belfort, Senior Medical Director of Praxis Precision Medicines. Dr. Belfort is a central nervous system, CNS, focused physician scientist and medical director. With 8 years of experience in the industry, Dr. Belfort is leveraging the latest advances in neuroscience to advance novel medicines through rigorous clinical trials. His work on neurosteroid GABAA receptor modulators contributed to the recent approval of Zulresso, brexanolone, for the treatment of postpartum depression. Now at Praxis Precision Medicines, Dr. Belfort is on a team utilizing the emerging genetic causes of various epilepsies for the creation of precision therapeutics for patients with these conditions. Dr. Belfort has conducted 2 successful studies at Nucleus Network and is working on the 3rd. Cameron Johnson is our next presenter, and Cameron Johnson is the CEO of Nucleus Network. Cameron is considered one of the most experienced phase 1 clinical trial professionals in Australia, having held senior management roles within 3 phase 1 Australian clinical trial companies. Johnson's phase 1 experience extends into Europe and the clinical research organization CRO industry, where he managed clinical trial facilities across Australia and New Zealand. He joined Nucleus Network in September 2013 as Chief Operating Officer before moving into the position of Chief Executive Officer in May 2016. Prior to commencing at Nucleus Network, Johnson was the inaugural CEO at an Australian Phase 1 clinical trials facility, Interstate. In 2019, Johnson led the acquisition of Q Pharm in Brisbane and Prism Clinical Research in Minnesota.
Excellent. Wonderful. Thank you for the invitation, Cameron, to speak on the important work that we've been doing together towards advancing our pipeline to patients with a variety of neuropsychiatric diseases who really, really need them. And thank you also to X-Talks and to Sonia for the kind introduction. So I'll just go on to the next slide, and this really provides a glance of the company. Praxis is a company that was initially started based off of the emerging findings in the field of the genetics of epilepsy. And I'll speak much more about that on the subsequent slides. But launched 3 years ago, we saw this field emerging and wanted to meet the leaders of it. And that's really what first brought us to Melbourne, Australia. The other important pieces of the slide are that we have quality investors. And if you look at the biotech blogs from about 2 weeks ago, you'll see we have our first press release out. And we're very excited to come out of stealth mode and really tell our story for how we're trying to develop new medicines to help people. On the right-hand side is a couple of little points. One is that our assets really are very different from one another. Some are small molecules, some are antisense oligonucleotides, and they really have are de-risked in the sense that if one molecule, if we find a problem with it, the other molecules are extremely unlikely to have the same, what we call problem. So we're ready to really launch this company in a big way, and we're really excited about it. And we've already been doing a ton of work with Nucleus, and I'm really excited today to tell you all about that. So just before we get to Melbourne and how we found Nucleus, I'll tell you a little bit more about epilepsy and the genetics of epilepsy. So we know, all of us know from our high school science classes that in 1953, Watson and Crick made their seminal discovery about the genetic basis of inheritance and the role that DNA plays in that. And really, in a short time, we've seen an explosion of understanding of the molecular basis of disease. And epilepsy is a fantastic example of that. So in just under 50 years, meaning in the late 1990s, we began to have the first examples of epilepsies that were tied to specific mutations in specific genes. And what followed from that was a series of sequencing efforts. At the bottom left of the slide, you'll see where it says Epi4K. Well, that was a sequencing effort to look at about 4,000 genomes, and that was actually led in part by teams in Melbourne, Australia. And so as Praxis began to be launched and the ideas for it and the funding for it came together, the obvious place to go to find out about these drug targets, these novel ideas for what we could develop new medicines for, was Melbourne, Australia. Say a couple more things about these targets. What you see on the left-hand side is in the purple ellipse are a series of genes associated with epilepsy. But you see that there's an overlap with psychiatric diseases, movement disorders, pain and migraine. And so what we know now on the right side of the slide is that those are all medicines that were originally developed for epilepsy, yet now have found utility in broader neuropsychiatric indications. So as an example, Depakote, top left there, that's a drug that's a very good anticonvulsant, but it's also used in bipolar disorder. Other medicines used for, again, migraine, pain, and movement disorders were originally developed as anticonvulsants. So start to put together a picture for what Praxis is. We think that these genes are super interesting because if we can develop novel medicines that target those genes, we think we can help the people with epilepsy. That's a great first step. And maybe an important second step is we take those medicines and help a broader group of people. So I'm showing you now here the leadership team at Praxis. Marcio Souza is our new CEO, and reporting to him is this fantastic executive team, including my boss, Bernard Ravina. He's a neurologist and the CMO at Praxis. And Stu Chafee is our chief business officer. For this talk, it's important to focus on the chief scientific officer and founder of Praxis, Steve Petru. So Steve Petru is one of the main reasons why we found ourselves going frequently to Australia and in particular Melbourne. Because Steve is not only the CSO at Praxis, he's also the head of neuroscience at the University of Melbourne at an institute called the Florey Institute. So I'll get into a little bit more depth later on, but essentially the Florey Institute and our relationship with them led to picking certain targets, finding medicines, and then we were in this position of, oh, this is fantastic. We've got assets that we want to bring into human clinical trials, but we really didn't know where to do that. Lo and behold, right across town, we found Nucleus Network. Fantastic collaborator, technically savvy, excited and hungry to do new things, and I'm going to tell you about a couple of those studies. So when we look at our pipeline, 114, 944, 562, 222, 020, all at different stages, is a very classic look for a pipeline at a biotech company. Some of the molecules are still preclinical, others are in healthy volunteer studies in phase 1, and yet others are in later stage patient studies in phase 2. What you see is a whole host of molecules. Some of them are small molecules, others are nucleic acid-based therapies. What I've circled here is our top 3 programs in terms of advancement into the clinic, and I've put this square around them because for all 3 of these, we've worked with Nucleus Network to try to advance our programs. I'll talk about these top 2 in more detail on the next slides. So Prax 114 is without question our lead molecule. We are developing it for the treatment of depression. And over on the bottom right here, I'm giving you an idea of why it's so important for us to be developing medicines for depression. 40% of patients with this condition are unresponsive to the standard of care. It takes a long time for those who are responsive, in fact, to have any clinical benefit. There's a lot of placebo effects, side effects of the currently available medicines. The long and the short of it is there's huge unmet need in the world, and I'll just speak very briefly that there's even unmet need in my own family. We need to have better medicines for these conditions, and it's a personal nature that I'm hunting for new medicines for this condition. So if we take ourselves over to the sequence of clinical trials I've laid out here, you see that right here is a major depression study in perimenopausal depression and major depression as a proof of concept study. So this is actually an ongoing study, but the predecessor study is what we conducted with Nucleus in healthy volunteers. This was a phase 1, multiple ascending dose study with biomarker work, and that's what I'm going to talk to you about in particular, in part because it's the more complicated aspect of the study, but also because it's my passion. I really am very deeply interested in translational biomarkers. But if you look at the study design, we had 3 dose levels. We had a 4th cohort where we studied food effect. These were healthy volunteers, 18 to 55 years of age, with no history or ongoing medical conditions. And then the main objectives of the study are to collect safety and tolerability and pharmacokinetics data, and to look at the metabolites of this drug in plasma and urine. But the third endpoint, and one that I'll speak to you about right now, is the pharmacodynamics, or the EEG endpoint. So what am I talking about? So essentially, EEG data, and some of you have seen this maybe on Grey's Anatomy or one of the other TV shows about clinical care, or maybe you've had one, but essentially we're collecting electrical signals at the skull surface. And why that's so important to us is because we develop medicines for the brain. And unlike, say, for example, hematological oncology drugs, where you can just take a sample of the blood and see how much drug is inside the cell you want to treat, or inside other parts of, or other cells that you're interested in, or floating around free in the plasma, or bound to protein, we can't actually access our tissue, right? We can't get samples of the brain to see how much drug is in there. So it's very important that we have surrogates. And in this case, what we were able to clearly see through our work with Nucleus is that as the concentration of our drug changed in the blood, in the plasma, we were able to see clear changes in the magnitude of a very specific EEG effect. And to be very clear, yes, this is a fairly undetailed figure. It has certainly been sanitized by our IP lawyers, but it was a critical piece of information for picking doses for the subsequent trial. So just to go back, the way we picked doses here was by knowing that they got into the brain and had a clear impact on brain function here. So we couldn't have done this critical study in patients without the data we had originally collected with Nucleus in this healthy volunteer study. So to move on to the study we are about to conduct, and this is really exciting. It's about another molecule, Prax 944. It's on track to be the first approved therapy in over 50 years for the treatment of essential tremor. A lot of people have essential tremor, as I'm showing you down here some of the epidemiology. Many of these people discontinue on the standard of care because of a number of side effects as well as lack of efficacy with what's available. Some people even go so far as to have brain surgery. Essential tremor is just what it says. It's tremulousness of the upper limbs. It prevents people from being able to eat normally, care for themselves, brush their teeth, comb their hair, and ultimately, a lot of these people end up living a fairly reclusive life because they're embarrassed by the tremor and going out in public. So, yeah, definitely a high morbidity condition, a lot of unmet need, and a place where we want to develop a new medicine. So towards that, we are conducting a study right now, which is showing some promise in essential tremor. And we're limited to 2 dose levels. And that's because when we first acquired the molecule, nobody had, the previous company that had worked on it had not really worked to understand how best to use this molecule. So there was some single ascending and multiple ascending dose data, but nobody had studied titration. And so that's what we want to do now. But it turns out that doing a titration study with this molecule, really with any CNS molecule, is very, very complicated. So let me take you through the design, and then I'll show you why the study is so complicated on the next slide. So part A, open label, we're going to explore a new drug product. It's actually just a 5 milligram tablet, when in the past we only had larger dose strength tablets. Part B, placebo-controlled, up to 22 participants, and a much more complex design. I'll focus on part B. There's a healthy volunteer study. In part B, we're going to look at safety and tolerability and PK of doses higher than were previously tolerated in a single ascending dose study. So how do we do that? So the hypothesis is if we basically slowly give you this drug, you're able to tolerate doses that are higher than if we give you a given dose on day 1. So what's tricky about that is collecting data and making decisions while you're conducting the study. So let me show you the design of the study. It's very complicated. We'll do some screening, baseline work to look at baseline EEG, and then we'll slowly titrate from dose 1 to dose 2, which we have extensive experience with, and now to higher doses where we have less experience, and higher yet as we progress through the study. Now, in order to make the decision that it's safe to go from dose level 3 here to dose level 4, we have to collect a lot of information: pharmacokinetic information, EKGs, laboratory information, even safety EEGs, and all of that needs to be rolled up in a way that's understandable in order to make a decision at something called an SRC, which is a safety review committee. And for each dose level, we've got to do that same cycle of work in a very short period of time in order to make the decision to titrate to the next highest dose. So as you can see, this is an ambitious, technically complex, and integrated effort. And really, we would only feel comfortable doing this study with Nucleus. So, and that's predicated on the previous experience we've had doing complicated studies, utilizing EEG, and because of the quality, safety, and tolerability data they've collected for us in the past. So with that, I just wanted to reemphasize our top 3 programs. We've worked with Nucleus to develop these molecules, and we feel confident and really excited about the prospect of advancing these molecules towards patients who really need them. With that, I'll say thank you and turn it back over to Sonia and to Cameron.
Thank you very much for that, Dr. Belfort. Now I'm going to pass over the controls to Cam. Cam, when you're ready, you may begin. Do you see it there now, Cam?
Yes, I see it show my screen. Fantastic, thank you.
Perfect, no problem. Thank you.
Excellent. Thank you, Dr. Belfort, for those kind words, obviously, and likewise, we're very much looking forward to the next collaboration with Praxis. Before I launch into the main sort of body of the presentation, and really to go through a little bit more in regard to what Dr. Belfort mentioned in terms of specialist capabilities within Australia, etc. I just want to provide you with a very, very quick 1-slide high overview of Nucleus, and then I'll get into more of the deep presentation then. So in short, Nucleus Network, we're a dedicated phase 1 clinical trials organisation in Australia. So in total, there's about 250 phase 1 beds available, and Nucleus Network is about 150 of those beds. And so in terms of the organisation, we've been in business since about 2002. On an annual basis now, we conduct around about 70 phase 1 studies. And of those 70 phase 1 studies, really about 30 to sort of 35 of those studies are that true first in human space. So certainly as I launch into the presentation and expand more about what Dr. Belfort mentioned, I'll sort of take you through some of the highlights as to why Australia is potentially punching above its weight in terms of the number of first in human studies conducted. And then also before sort of I launch into it, I just wanted to provide the audience with, some assurances that, we've had multiple FDA inspections, we've had EMA inspections, previously had and visa certification. So certainly from, a global gold standard type of quality, we're very, very familiar with various regulators and expectations of them. So really launching into the presentation from here, this presentation will focus on 4 key elements, that were drivers in not only Praxis's decision as to why to conduct phase 1 studies in Australia, but also more broadly, while the USA, Asia, and an increasing number of European companies are looking to place their phase 1 work in Australia. Quite simply put, there are 4 key elements that are the drivers of Australia. When we look at speed into clinic really is that major driver, that key milestone that companies want to achieve. And that's where Australia does really offer a quite expedited path to get into your sort of first patient, first dose. Then from a quality aspect, I'll take you through how foreign regulators view the data generated. From a cost aspect, how does Australia stack up against other countries? And obviously quite interesting with the COVID situation these days as to sort of how the Australian dollar has fared against other currencies in the world. And then really one of the aspects that I will take a bit of a deeper dive in is really that specialisation. So obviously Dr. Belfort mentioned his keen interest in PD measurements and really can sort of expand upon that a little bit further in terms of what does the clinical trial ecosystem look like in Australia, for supporting not only sort of basic type PK studies, but what other more novel, unique PD biomarkers can you potentially put in to your programs and have them executed. So when I look into speed, so to be honest, the number one reason particularly USA companies come to Australia to run clinical trials in phase 1 is that you don't need an IND to start a trial in Australia. However, I really do want to stress that the preclinical data package that is required in Australia is identical to that of the USA. So certainly there's no shortcuts in terms of the preclinical, very high level, there'd be an expectation that, naturally you've got 2 species, either 14-day or sort of 28-day repeat dose studies are completed. However, the interesting part in Australia really is that pivot point, is that once you've got a final protocol and once you've got draft reports from your preclinical studies that can go into your investigator's brochure, you're essentially good to go in Australia. So you've got the protocol, you've got the investigator's brochure, and then it's those 2 key documents that get submitted in Australia to the IRB. And you can certainly see here on sort of the right-hand side of the screen that in the Praxis 114 study, you can see there that from the date of submission on the 2nd of May, the actual ethics committee reviewed that package essentially just over sort of 3 weeks later. And then within 10 days of that meeting, we actually had the ethics approval in our hand. And then, so that was essentially, you can see that about a 4-week path from time of the documentation was submitted to time the documentation was approved. And then in this particular program, we had around about a 3-week screening window. So to obviously get the SIV done, drugs in the country, and then to have that first patient, first dose completed then on the 24th of June. And then just some other sort of brief points when we look at CMC and speed is that there is no separate dossier of CMC information that's required to run a study in Australia. So your CMC package is really as brief as to what the ICH guidelines require in the investigator's brochure, which we've seen as little as, a page to sort of half a page, which is obviously very unlike IND applications and the amount of CMC rigour that's put into those programs. And it's also worth mentioning, although we don't see it on a lot of occasions, and this guidance is the same as in the US, but it's actually not mandated that you need GMP material to conduct phase 1 studies. Obviously, GMP materials are absolutely needed for your phase 2 and your phase 3 programs, but certainly when you're looking at a phase 1 program, it's the regulator state it's not essential that it is GMP certified. Obviously needs to be manufactured to the principles of GMP. It doesn't need that final sign-off and documentation. And then just finally on speed, wanted to sort of take you through, how does Australia get approval for a clinical trial within 4 weeks without an IND application, really on draft reports from your investigator's brochure. And it's basically the system in Australia for clinical trial approval is called the CTN path. And basically that CTN path is known as the clinical trial notification. So the Australian regulator is called the Therapeutic Goods Administration, or we like to abbreviate as the TGA, which is naturally the equivalent of the US FDA. And within Australia, our regulator has said that the review and approval of a clinical trial in Australia is the responsibility of the IRB or Ethics Committee. I think we use that language sort of interchangeably here in Australia. But essentially the Ethics Committee is the overall reviewer and approving body of the trial. And once we have that approval, we merely need to notify our government that the clinical trial is due to commence. So our government does not review protocols, it does not review investigators' brochure, it does not hold the process up. It's basically just a notification that Ethics Committee X has reviewed, approved it, this is the approval number, and then our government just logged that clinical trial is due to commence in Australia. So certainly a very, very simplified path in terms of it. Certainly it's a very kind of rigorous path. So as I mentioned on the previous slide, it was about sort of a 3-week review process. And within the ethics committees in Australia, you do have a scientific subcommittee. So you've got sort of both committees that work in parallel. So the scientific subcommittee issues questions that you would expect from a regulator. They'll very much be looking at the starting doses, they'll be looking at the dose escalation cycle, they'll be looking at the preclinical studies, if there were any safety signals, and then they'll issue questions. And then you've obviously got the ethics committee, which is very much obviously looking at the participant information and consent form and obviously dotting your I's and crossing your T's. So I just want to really reiterate that there is a scientific review done that's similar to that of a regulating body, but it's actually just wrapped up in the ethics approval process in Australia. And then from a quality aspect, so if you've made a decision that Australia looks like a favourable destination, it's great that we can get into clinic quickly. What do other jurisdictions think about the data that's being generated? So we can certainly say that I sort of alluded to at the start of the presentation, was that Nucleus specifically, we've had 3 FDA inspections. We've had no 483s issued from them. We've also had an EMA inspection, and then previously we held an visa certification. Also probably want to mention on that, it's a little bit unusual for phase 1 facilities to get FDA inspections, given that we're typically not generating pivotal data in the registration package. However, in the 3 FDA inspections we were involved in, one was for 505(b)(2), so obviously pivotal data we generated. One of the inspections was triggered for an ANDA program, and then the final inspection that we had, which was in 2018, was for a biosimilar program where we were the lead recruiting site for that. And so with the biosimilar program, we were on the FDA radar as pivotal data to that registration, and also the EMA came out to look at that biosimilar program as it was going through the registration path there. So in terms of strategies, so what typically do we see? Companies come to Australia, they can get into clinic very, very quickly. They can get the phase 1 PK, hopefully there's PD biomarkers in there, obviously that Dr. Belfort mentioned in some of the Praxis programs. And then typically we find that companies take that information, the data they generate in Australia, they take that back to the US, and then they open their INDs directly into phase 2. We certainly have never had an instance where a client has gone to the FDA with data that we've generated and there's been any questions or concerns where it's needed to be repeated or anything like that. We find that US biotechnology companies, they really see Australia as that leap into phase 1 studies, but then naturally with our limited population, Australia's got a population of around about 25 million, that we don't necessarily have the population to support, standalone country phase 2 programs. So therefore it makes sense to open the IND, get sites in the US. Often Australia may be a site in the phase 2 program, but rarely is Australia looked to as the sole recruiting country for a phase 2 program. And then sort of finally, in terms of sort of 3 high level, I said speed, quality, and then we look into cost. So certainly with the COVID situation now, Australian dollar has taken a little bit of a hammering there. And so we used to say that, head to head, Australia was around about sort of 20 or 25% cheaper than the US from a head to comparison. But certainly over the last couple of months, the Australian dollar has slid considerably, and so now we're sort of operating at around about 65 cents to the US dollar. So based upon sort of exchange rates alone, Australia is 30 to 35% cheaper than what you would for running a clinical trial in the US. And then in addition to that, there's the R&D rebate system that's certainly been around for some time now, where essentially for every dollar that a company spends in Australia on R&D activities, so that includes the clinical trial, it can include manufacturing, it can include your data management, stats, medical writing costs. So for every dollar that you spend in Australia on R&D, such as a clinical trial, the Australian government is actually giving 43.5 cents back in the dollar. So certainly when you put sort of the 30-35% savings from exchange rate, say if we call that sort of a 40% saving from R&D, Australia certainly is a much cheaper destination, say than the US, to actually can conduct studies. And then certainly what just on the side here is that there obviously is a requirement that if you are looking to access that 43.5 cents back in Australia, you actually do need to have an Australian subsidiary established. So certainly when we look at Praxis Precision Medicines, their obviously main office is in Cambridge, and they've set up as a subsidiary in Australia. And really this is no different to sort of around about 50% or so of our US clients are actually setting up subs in Australia to obviously take advantage of that 43.5 cents back. But we're also finding, you know, from an Australian aspect, it's certainly been very good for the economy. There's a lot more clinical trials being based here. And certainly companies are looking at placing multiple programs here as opposed to sort of one and then they go back to the US, multiple programs coming through. So then, as I mentioned sort of at the start, and you know, what I wanted to spend a little bit sort of time focusing on was really more that specialisation that Australia offers. So when we look at sort of Praxis Precision and what Dr. Belfort mentioned, you know, really having access to some complex PD markers, you know, was really quite advantageous. Obviously, it was mentioned that there's some great research institutes that are in Australia, and obviously specifically Melbourne in the Praxis example. But there's also a very, I guess, different sort of clinical trial ecosystem here in Australia to what you might find in the majority of places in the US. And really specifically, I wanted to focus on the location of the phase 1 units. And this is not just Nucleus, this is pretty much the majority of the phase 1 units in Australia, is that they're actually co-located within major tertiary teaching hospitals. So when we look at our Melbourne facility, we're actually physically in the building of the Belfort Hospital, which is an 800-bed tertiary teaching hospital. And then our Brisbane facility is physically co-located really in the Royal Brisbane and Women's Hospital, and that's a 900-bed tertiary teaching hospital. And really, there's a few aspects that I want to draw out from the location. One is safety. So certainly with the number of first-in-human programs that are being conducted, we certainly do see anaphylaxis. We see anaphylactoid type reactions occur in programs, and certainly knowing that you're supported by the hospital medical emergency department, where we can just pick up the phone, call the code. We have the crash team. The quickest I've seen the crash team there is in about 3 minutes, but typically it's about 5 to 7 minutes that we would have the emergency response team. And then we're also able to transfer directly into ICU, CCU, or the emergency department, depending on what the emergency may be. So certainly from a safety aspect, we find that that's very critical. And then some of sort of the other softer parts that are certainly very valuable, though, is that we also get access to hospital clinicians. So certainly in the examples of Praxis studies, is that we've worked quite collaboratively with the hospital neurology department and sort of some of the KOLs that are based there. And then obviously having access to hospital clinicians, we also get access to patients as well. So, and I'll expand upon that a little bit more. And then finally, also by being in a hospital, is that we're potentially able to access quite complex and potentially invasive PD assessments that may not have been able to be conducted, you know, where you were phase 1 unit sort of in an industrial park or in an office building. There's certainly some advantages from a PD aspect. So certainly when we look at the hospital clinicians and patient access, we find that a large number of the phase 1 first-in-human studies that we conduct, we're able to run adaptive protocol designs. So the way I refer to as an adaptive protocol design is that start your single ascending and your multiple ascending dose in healthy volunteers, but then actually able to put cohorts of patients into that program once you've reached an appropriate dose level. So certainly there, you know, this system has worked really, really well where, you know, we've done respiratory programs where we've been able to then recruit IPF patients. We've done programs where we've been able to recruit NASH patients. Alzheimer's disease patients have also been recruited, hepatitis patients, psoriasis, mild to moderate asthmatic patients, COPD, and also diabetes as well. And then I've also mentioned sort of some of the other therapeutic areas that we've been involved in. where we have worked with hospital clinicians, obviously experts within that therapeutic area, been able to provide great guidance in the first in human program, and then also to be able to put patients into that program, single protocol, Nucleus as a single site, certainly has added a lot of value to our clients. And then when we look at sort of the PD aspect, so if we take the Praxis example, some of the really, I guess, cool, unique sort of PD biomarkers we've been able to put into programs. You know, one of them has really been around sort of those EEG measurements. So obviously working with the neurology group for training, for placement of the electrodes, etc. That's been quite advantageous. We're doing a lot of work in the CNS drug development space. So we're finding that we're doing more and more lumbar punctures on healthy volunteers, the CSF collection. So working collaboratively with the hospital anaesthetics department. We've run some first in human programs where we've done 3 lumbar punctures on healthy volunteers. So one at screening, one just after dosing, and then another one about 14 days later. So certainly these lumbar punctures are done within the clinic and done obviously by very experienced anaesthetics doctors. And then sort of to go a little bit further into some of the unique biomarkers from a respiratory aspect, we've also done bronchoscopies on healthy volunteers, so working on inhaled product where we could then transfer the participants into the bronch suite and then do a bronchial lavage on the healthy volunteers for some PD biomarkers. And then also been involved in some gastroenterology programs where we've actually been able to do sigmoidoscopies on healthy volunteers as well. So, you know, you can really see that we do work quite collaboratively, really kind of leverage that relationship that we have with the hospital, and so that we do get access into, you know, really some pretty unique and cool kind of biomarkers. And then in addition to that as well, is that we are able to do sort of PBMC. So if you look at the immunology space, being able to run PBMCs on site, flow cytometry on site, cytokine assays as well, where you've got fresh samples that are time critical. There's certainly those capabilities within sort of the grounds of the Nucleus facility. And then more broadly, sort of as I go to wrap up the presentation, so more broadly, it's not just sort of around that first in human space. And as I mentioned, although that is really sort of 50% of the work that we do, and obviously with Praxis, working in that kind of SAD, MAD type design space, but certainly, you know, we probably capture the full gamut. We've announced the other week, and we've got dosing, you know, shortly, where we'll be running sort of a COVID-19 vaccine program for Novavax, who's a NASDAQ listed company. So obviously very excited to be involved in that program. There's a number of other COVID-19 vaccine studies that we're also involved in, where announcements will be made shortly on those as well. And then sort of, you know, some other more standard traditional phase 1 studies, your bioequivalent studies, biosimilar studies, thorough QTC studies. And probably what some people don't probably realise from Australia also is that we are a very multicultural country. And so certainly access to Japanese, Chinese participants is certainly possible. So where we've been involved in sort of ethnopharmacology studies, you know, for about the last 10 years or so. And then sort of finally, as I mentioned, that really the biotechnology sector is really where they see the real value in coming to Australia, getting into clinic quickly, using that data to support an IND, obviously achieving milestones much earlier to release more funding into companies. And, you know, while some might raise more money for development, others will look for an exit after their phase 1. So certainly commercially, we find the biotech sector very much value what Australia can do. And then from a client base, we certainly find the majority of the work is based from US companies, but we're seeing an increased presence of Asian companies coming to Australia, and certainly European companies are looking more and more to Australia, you know, and particularly with the COVID situation as well. Australia has fared very, very well in terms of the number of cases. So certainly seeing, a number of clients that are moving programs from Europe or that were up in the US to Australia, just given the, essentially very safe climate that we have as it relates specifically to COVID. And then just sort of finally to wrap up, so 4 to 5 week approval process, that's governed by the clinical trial notification scheme. Certainly specialising in sort of that first in human space, data is acceptable, sort of the FDA, EMA, and certainly companies use Australian data alone to support their packages as they open their INDs and then move into phase 2. Obviously, I just like to thank Dr. Belfort for his participation. Obviously, thank X-Talks as well. And then following the presentation, there's certainly an opportunity to ask questions. And yeah, thank you very much for your time.
Well, thank you very much, Cameron, and thank you very much, Dr. Belfort. Thank you very much for that very insightful presentation. I hoped everyone enjoyed it. Now, I would like to invite our audience members to continue sending in their questions or comments right now using the questions window for the Q&A portion of the webinar. I've already received a couple already, so I will start with those. So, Dr. Belfort, this question is for you. Is this an inpatient study, is what the audience member is asking.
I appreciate you sending that my way. The studies we've conducted with Nucleus have been exclusively inpatient, and I think you may be asking that because the 944 study I described is certainly a very long study, but yes, that will be, Part B is in fact all inpatient.
Okay, thank you. Don't go anywhere. We have another one for you. Dr. Belfort, what work led you to the discovery of the EEG effect?
So the EEG effect of this particular class of molecules is pretty well established. We have said publicly that these molecules are GABA A modulators, and if you search GABA A modulators in PubMed or any other large publication database, you will come across EEG signatures of that type of molecule.
Okay, thank you very much. The next question I have is for Cameron. Cameron, please decode the TGA.
Yeah, certainly. So the TGA is called the Therapeutic Goods Administration. So the Therapeutic Goods Administration, is the governing body of Australia. So the US has the FDA, Australia has the TGA. So it's the Therapeutic Goods Administration.
Thank you very much for that. Let me go to the next question I have here for you. This is for you again, Cameron. We are looking for phase 1 dose expansion sites. Will that be of interest to Nucleus Network?
Yeah, certainly. So we do have sort of a dedicated oncology business unit. So from sort of dose expansion, we find that, we are involved in a number of oncology programs where sort of we involved in expansion cohorts there as well. So certainly, interested in following that up after this presentation.
Okay, thank you very much for that. The next question I have is, in addition to Nucleus, are there support services that facilitate trial execution?
And who did you want to answer that?
Either one.
I mean, certainly Cameron's the expert on the various services around, but I can tell you in the studies that we've conducted in Australia and in particular with Nucleus, there have been outstanding support services in the form of CROs to help conduct the studies, to do analytical work on the samples obtained in the studies, as well as local companies that manage the trial drug.
Okay, perfect. Here's the next question. This is for you, Cameron. Does Australia have any concerns or restrictions with testing immunosuppressive drugs in healthy volunteers in the current environment? Immunosuppressive drugs.
Yeah, no, really good question. So Certainly on that, there's no sort of government restrictions as such. It's more a site-specific sort of chief medical officer, principal investigator discussion. So short answer is that it's assessed on a case-by-case basis. Certainly there are some first-in-human programs we're running that are involved with immunosuppressive drugs, but certainly we're comfortable we're at the start of the single ascending dose, so very, very low single dose, where there's sort of no thought or literature to support that a single dose would be immunosuppressive or has a very short half-life and the participants are actually confined. But however, we certainly have got some other immunosuppressive drugs, some immunosuppressive programs that are up to the multiple ascending dose, and they've been paused for the minute. So on that note, no really clear straight answer. It's every study is being assessed really on its own merits, and that's a discussion sort of with the Chief Medical Officer of Nucleus and also the company. But we certainly are running some programs with immunosuppressive drugs on healthy volunteers.
Okay. Here's the next question. Have you run later phases studies in Australia?
Look, not Nucleus specifically, other than, well, we do take on some phase 3 vaccine programs where due to the infrastructure that a phase 1 facility has, to be able to recruit sort of 200 or 300, 400 people very, very rapidly can be done. But typically, clinical trials that are outpatient-based only aren't necessarily kind of Nucleus Network's sweet spot. We're very much about confinement. So as I mentioned, sort of we've got 150 beds in Australia, and so the majority of the programs that we're involved in either require a confinement period or alternatively are very, very high volume studies such as vaccines.
Okay, thank you. This question here is for Cameron. Have you conducted phase 1 healthy volunteer, phase 1B patient hybrid studies? And if so, how do you identify and enroll neurology patients?
Yeah, certainly. So on the short answer is yes, absolutely. We do quite a lot of hybrid studies. I think the terminology that we often use is more about adaptive protocol design. So obviously healthy volunteers in your single ascending, multiple ascending dose, hopefully, get to the dose level that you want to explore further, expand that out into patients. And so how we recruit the patients is that we work sort of with network of clinicians that refer into Nucleus. So probably a good example, say is the oncology programs. And so our oncology team, they get a lot of other oncologists around the state of Melbourne or Brisbane to actually refer their patients in. And certainly when we've been involved in sort of some Alzheimer's disease programs previously, is that we've essentially had a network of about 4 or 5 referring physicians. So they look through their database, they refer the patients to Nucleus, we do the consenting screening, and then enroll them from there. So certainly the adaptive or hybrid study design is something that we're very familiar with. The ethics committee is very, very familiar with it as well, and we certainly don't have any issues from a single protocol approval process.
Okay, perfect. All right, I have a couple more questions I'm going to try to squeeze in. So here's the next one. Has the COVID-19 pandemic impacted recruitment of participants?
Yeah, I'll answer that one. It's actually done the opposite. So we've actually seen a huge influx of trial participants coming in. And, that's really around, obviously, with, so many restrictions put in place. Obviously, the hospitality industry is obviously, very much been decimated by COVID. We've actually found that we've got, almost a new group of participants wanting to do clinical trials, really because of the climate. We're also finding that, the people that turn up, the people that are enrolled in trials are much more committed than what we had seen previously. So certainly we've seen a positive effect on recruitment due to the COVID-19 pandemic.
Wow, thank you very much for that. Here's the next question. Is it difficult to transport samples to the USA or EMA for analysis? And does this impact study timelines?
Yeah, so it's Cameron. So on that, typically, so we certainly ship samples to the US and Europe regularly. We do multiple shipments per week, and typically we're not finding that's rate limiting. It's fair to say, probably for most of the first in human programs that we do, bioanalytical analysis is often done in country. But certainly we have shipped first in human samples overseas before and still been able to sort of dose escalate around about that sort of 10 to 14 day turnaround for a single ascending. And obviously when you look at sort of biosimilars, TQT studies, etc., where you can kind of batch ship samples, there's certainly not been any issues with shipping samples to those destinations, and we've shipped into China, Japan, etc. before as well.
Wow. Okay, great. All right, we have time for just one last question, and here it is. How long in advance does Nucleus Network need to guarantee bed availability?
Yeah, so on that, we typically lock our beds around about 3 months in advance. And so kind of when you go through the timelines, we say that, and that's 3 months from first patient, first dose. So if we kind of work our way backwards, I said it was around about a 4, 5, 6 week approval process.
Mm-hmm.
So obviously you've got your 6 weeks for your approval, and then we like to be engaged, at least sort of 6 weeks or so before we're submitting documents to the ethics committee, just so that our medical team, operations team have a chance to sort of really review that data, provide input into the protocol. So really, say the shortest is around about that 12 weeks from your first patient, first dose. But obviously, the more notice that we're given, the better for everyone.
Okay, thank you. Well, thank you very much for those questions. We have reached the end of the question and answer portion of the webinar. If we couldn't attend to your questions, the team at Nucleus Network and/or at Praxis Precision Medicines may follow up with you after this presentation. If you have any further questions, please direct them to the email address on your screen, and that's to Cameron Johnson, so that's c.johnson@nucleusnetwork.com.au. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to this recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues when they register for this recording as well. So I encourage you to do that. Now, please join us in thanking our speakers, Cameron Johnson, CEO, Nucleus Network, and Dr. Gabi Belfort, Senior Medical Director of Praxis Precision Medicines, for that very insightful presentation. So thank you very much, gentlemen.
Thank you.
Thank you. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, take care and bye for now. At this point, I like soda.